time to learn about how we're going to take genetic information to use that to code for the expression of traits. So just some reminders, DNA contains the instructions to produce proteins, which are our traits. Those instructions are to make our traits, but proteins are produced at the ribosome, which is outside of the nucleus. So how does it happen? Well, it's going to use the help of a friend. So just kind of giving you an overview of this image, you have DNA inside of the nucleus of the cell. You can see it's kind of labeled here as the nucleus. And you're going to see the word synthesis again. It means to build, and it's going to build a copy of mRNA in the nucleus from the DNA. So that's why it's called messenger RNA. It's taking the message then out of the nuclear pore, out of the nucleus, into the cytoplasm, going to a ribosome. So that movement of mRNA into the cytoplasm via the nuclear pore, and then the synthesis of a protein. So the next thing that's built is the protein, and you can see that the messenger RNA goes through the ribosome and amino acids are built. The next step after this is that we just want to review some of the players involved. So we have DNA, messenger RNA, ribosomal RNA, and tRNA. These are all of the molecules that are involved in building a protein. So the first phase of protein synthesis is called transcription. Your genes are a code to produce a protein. So we need to get from DNA to mRNA. So what has to happen first is the DNA strands have to separate because of an enzyme called RNA polymerase. Now this may sound familiar because we talked about DNA separating whenever DNA was going to make a copy of itself, but this is different. Now DNA is going to separate because an mRNA molecule is going to need to be created. So again, this is an enzyme RNA polymerase. The new strands are now template and complementary strands, and this takes place in the nucleus. mRNA is going to match up with the template strand. So here's an image of what's happening. So here's your DNA. You can see it's unwinding, and you see that this red is going to be the mRNA that's being made. So in the image below, you'll see terms like promoter, terminator. Those aren't important right now. That's a little bit more detail than we're going to get into. But you can see that the mRNA is growing as the different nucleotides are added. So let's take a look at this image. It just shows a little bit of another computer animated image. You see the promoter region, which is kind of really where this all initiates. Here, this oval shape is RNA polymerase, so that's what's going to start to, you can see down here, unzip the DNA. And then you have another molecule then that's the polymerase is going to work along here to make a copy of mRNA. Now, one thing that you may notice is that you're starting to see a U in the mRNA compared to in DNA you see a T. So we have some new rules here. There is no T in RNA. So when you go from DNA to DNA, our base pairing rules were A and T, T and A, G and C, C and G. But when you do transcription, which I have here, when you go from DNA to mRNA, the base pairing rules become T pairs with A pairs with U, C pairs with G, and G pairs with C. So right here is the difference. So this is a strand of DNA. This is my attempt at kind of modeling this for you. So this big thing here is the nucleus. This shape is RNA polymerase, okay? And this is our DNA strand that we're going to copy. So now we're going to select the correct nucleotides to join in creating an mRNA strand. So T pairs with, I'm going to drag an A. Let's see, I think I might have to. All right, so we're going to take T and drag it to A. A pairs with, Hopefully you're saying U, because remember there is no T in mRNA. C pairs with G. C pairs with G. T pairs with an A. And G pairs with a C. So now we just made our complementary or mRNA strand that reads AUG GAC, and that is now our mRNA strand. That wraps it up for transcription. Now what's going to happen to the mRNA molecule is it's going to leave the nucleus through the nuclear pore and take us into our next step of protein synthesis, which is translation.